Okay, welcome to Episode Genetics. I'm your host, Daniel Crossman, and this is episode 2. Last time we talked about acute lymphoblastic leukemia in very broad terms, causal factors, risk factors, stuff like that. This time we are going to go more into what is happening on a molecular level in this cancer. First thing I need to do, though, is introduce you to Peter Kramer. If you're like me, you didn't know who Peter Kramer was, but he is a very important guy. He just so happens to be the president of Universal Pictures. You know, that big, important, lucrative studio company. Yeah, you get the point. He's important. Anyways, let's say that you and Peter are sitting in a boardroom, and Peter's thinking, and suddenly he comes up with an idea. And he turns to you and he says, "Hey, I read a book, and I want to make a movie. Because, we're a production company and we do that so we can make money." So he sends you on your way to go to your kind of crappy little office, and he says, "Get this thing going." So you call Steven Spielberg and you say, "Hey, you want to make a movie?" Steven says, "Okay, I'm in." And from there, he starts putting together a cast. "Jeff, you want to make a movie?" "Yeah, sounds good." And slowly yet surely, you get everybody together. Yeah, Samuel, he wants to make a movie too. But it takes a lot more than just a cast and a director and an idea. You have to have cameramen and lighting and audio and editing, and well, we'll just call this production. It takes a production team too. So to recap, Peter has an idea. He sends you on your way to talk to Steven, to put a cast together, to work with production, to create a movie. Hey, you did it. And it's a great movie, and it makes lots of money, and people like it, and good job. You did it right. But the real question is, what the heck does this have to do with genetics? And that's a fair question. It's at this point that I would like to introduce my paper. Many papers on acute lymphoblastic leukemia are overviews, like this is what we know about the cancer. So here's a bunch of information. And I would like to focus in on this paper, which can be cited in the show notes and in the comment section, on the primary abnormalities that are happening. And specifically, we're going to go into the lower part of this figure on the left, B. So we need to talk about transcription factors. That is something we talked about in class. If you happen to remember them, they direct transcription. So of DNA to mRNA, they influence the rate. And they do this by binding to a specific DNA sequence. So they can more or less kind of turn on or off the gene. They can increase transcription. They can boost it. They can decrease transcription. They can repress it. And both are indicted in cancer. So good old Pete here, he's our TEL gene. And TEL is really important in progenitor cells in the bone marrow. Again, acute lymphoblastic leukemia is a leukemia, a blood cancer. Progenitor cells are kind of like stem cells. They can differentiate into different cells, different blood cells in this example. But they can't divide as many times as stem cells. So we have you in this situation is AML1. And AML1 is really important. It's a DNA binding component. And both of these, it's a part of a core binding factor is what it's called, along this transcriptional pathway. Both of these are really important in hematopoiesis. Let me try that again. Hematopoiesis. And that is just the normal production of blood cells. Okay, so our director is the MLL gene, mixed lineage leukemia protein. It is really, really important in expression, in regulation of what's happening, of like cell growth and self-renewal. It maintains expression of particular family members along the Hox family pathway. HOX is, you've probably heard of it, homeobox, super important. There are actors here. And everything is actually playing on this transcriptional HOX pathway. HOX genes don't necessarily give the information on how to build something, but they tell downstream when to build something. Really hugely important in development. And so finally, our PBX1 is it acts on the HOX pathway, and it actually represses. Remember, this is a normal pathway. So in a normal healthy pathway, PBX1 is actually repressing when we're making new blood cells. So to recap, our TEL, our president, gets with AML1, and together with MLL, they act on the HOX transcriptional pathway, which can be acted on by PBX1 to create a movie. But things don't always go the right way, as was my case, where you can have two genes fuse together. And when that happens, you get fusion genes. And these genes still work, and I'm saying work in quotations, in that they will create a protein, but that protein is not exactly doing what it's supposed to do in the way it's supposed to do it in a healthy way. So we have our TEL AML1 fusion gene here. And this can happen with the MLL1 too. It gets a fusion gene as well. And I should mention with the TEL AML1 fusion gene, it will actually inhibit normal transcriptional activity. So it changes transcription. It changes that whole regulation process. Here, it's a gain of function. And so it just wants to pump more and more and more out. And it enhances transcriptional activity. So again, changes in self-renewal and growth of those cells. So already our transcriptions are off. And then with PBX1, that is also, it's indicted or involved in a T119 translocation, which creates another fusion gene, the E2A PBX1, and that again disrupts normal expression of the HOX genes and other transcriptional factors. So it's just chaos at this point. Everything's not doing what they're supposed to. It's not looking like they're supposed to. And the HOX transcriptional pathway is like, what am I supposed to do? What's happening here? And so we have these genes that come together, the fusion genes, and they pump out something they're not supposed to, and they're changing the transcription amount. So does that sound familiar? Your cells are getting out of control, doing things they're not supposed to. They're not programmed to live this long or to produce this many. And your HOX pathway doesn't know what's going on. And anything acting on that HOX pathway is like, what is happening? And you go from a normal pathway represented on the bottom here, where in red you can see stuff acting on the normal pathway, all the way down to those target genes. See how it's all downstream. You go from making something healthy and good to something that should not be made. And you know what the problem with cancer is? They just keep reproducing cells over and over and over and over and over again. So I know that was pretty quick, fast, and furious, for lack of a better term. But hopefully it gives you a bit better of an idea of what's happening along at least one pathway when acute lymphoblastic leukemia sets in. Of course, we have a lot to learn and a lot of other areas that we can look into as well. I hope you enjoyed it, and we'll see you